The approval process for biosimilars is really interesting. People usually show a slide that has a triangle, and there's a triangle that is upside down, right? So there's a point and then the triangle, and the big part is the top. So it's like an upside down triangle. And the bottom is sort of as you're developing the biologic agent and you're doing some purification processes. And of course that takes a lot. It's kind of, it looks like it's the point of a triangle, but it's a huge amount of work, right? But then you go to the really meat of the whole matter, which is showing that these agents are safe and effective in a clinical setting. So you have to show not only your original clinical setting, but then you have to test it in all others. So a nice example of that is trastuzumab. It took a long time to develop trastuzumab and show it was safe, but then you start doing clinical trials. You give it in one trial with one combination, another, show survival is better, move it into early stage cancer, give it in combination with different targeted agents. We're still using trastuzumab as a backbone for studies looking at ways to expand the treatment options and improve outcome for patients with HER2-positive breast cancer. So the clinical part is the biggest part of the triangle because it lasts for a long time and the drug is tested in many different ways. So that's the reference biologic. When you get to the biosimilar, the triangle is reversed. So that reference biologic has done all of that work at the top of the triangle, and the biosimilar is really showing that it's biologically similar. So the triangle is reversed in that the need to show that it's similar clinically is really sitting on top of a much bigger weight of evidence that shows that it's similar in the laboratory and biologically. So you have to show that it has a similar structure, which is a complicated process because these are antibodies made in living cells. And you have to show that there is similar biologic characterization. Now, the caveat to that is that we know that any biologic test that you do in vitro doesn't correlate very well with what the agent does in vivo, so in a patient. But there are still tests that are done. One example is antibody-dependent cellular cytotoxicity, or ADCC, is something that can be done in a laboratory, and you can at least use that as a way to compare and contrast the reference and the proposed biosimilar to see if there are major differences. But that, that's not the end all by any means. And then you look actually at a whole lot of other structural characteristics. So you look to see, for example, if there's a difference in the number of sugars that are on there. It turns out that the differences are mostly in the amount of glycosylation, which doesn't seem to make a big difference, actually. You look in animal models at pharmacokinetics, pharmacodynamics. You look at immunogenicity in animal models. So you can imagine the bottom of the triangle is really the big part where you're doing lots and lots of studies, and many more than even the reference biologic was required to do to show that it's biologically similar. And then the very top of the triangle is our clinical studies. And the idea here, and as defined by the different regulatory agencies and World Health Organization, is to show that there are no biologically meaningful or clinically meaningful differences between the reference product and the proposed biosimilar. And so you have to have shown all of that sort of pre-work, working in the laboratory, in the clinic. Then when you go into people, what's been interesting is that, through the initial work of figuring out how to do the clinical studies, the idea came up that, did you need to show that if the reference biologic was made in the United States versus the EU, were they different products? And it turns out that when you move the production of a reference biologic, or you simply change the production process, you can change the reference biologic enough so that it is actually as much change as a biosimilar would be, because where you make the biologic and how you make it really makes a very big difference when you're making something in living cells. So one of the things that was needed to be done, for example, for the biosimilar trastuzumabs, is in healthy subjects, not people with cancer, but in healthy subjects, you had to show that the pharmacokinetics and pharmacodynamics were similar, obviously very short exposure, comparing the proposed biosimilar to EU and US trastuzumab, because the two of them could be different. So very, very interesting, because we hadn't thought about that before there were biosimilars in the clinic. We never thought about that. There's also been some really interesting work that has shown that actually some of the potency of ADCC, this antibody-dependent cellular cytotoxicity, may vary Not that it affects the clinical efficacy, we don't know, but from lot to lot of produced trastuzumab, there may be variations in ADCC on the reference biologic, which is also quite interesting and has been looked at in some of the reference biologic used as a control in the biosimilar trials. So quite intriguing. So basically you have to show all of these things are in the sort of preclinical area and healthy individuals are the same. And then you get to the actual efficacy and safety in patients. And what the regulations have noted is you want to choose a area to study your biosimilar that is a clinically sensitive area. So, for example, you would go back and you would look at the development of that reference biologic and you would say, where have we seen a big and reproducible impact from that agent that we would trust that it would actually tell us the agent works? So that's brought up some controversies and it's differed based on the agent, et cetera. But in general, you pick a sensitive endpoint, clinical endpoint, and then you, sensitive area, so indication for the drug. But then you have to pick an endpoint. So for most of these reference biologics, when they got approved, they needed to show an endpoint of disease control and for many of them overall survival if they're cancer drugs. A little bit different in the rheumatologic area and for supportive care where we also have biologics. But if we talk about cancer drugs, it's been really showing efficacy and the reference biologics, in many of them, have actually shown survival differences. So if you made the proposed biosimilar do the exact same thing, you would never get them on the market. That's part of this whole idea of competition, is that you want to use a short-term clinical endpoint that correlates with the long-term endpoints and use that as your approval. So basically, response rate has been used as a short-term clinical endpoint with the idea that in most cases, although you're not obliged by regulatory guidelines, you would look at the longer-term endpoints over time if you're able to. But every time you extend out your study, you add cost, right? So if you're going to follow people for 10 years, that's going to be a big cost added to the development of that agent. So then you have potential approval, right? You've done your clinical study, you've shown that your short-term endpoint is similar, maybe it's response rate, either in the, for example, for a cancer agent like trastuzumab in the neoadjuvant setting or in the first-line metastatic setting. So then the question is, what do you do with that data? And so that comes up with two other areas where the regulators have had comments, and one is interchangeability and extrapolation. So if we talk about extrapolation, the idea is if these biologic agents worked just as well in combination with different chemotherapy drugs, with different hormone agents, in combination with different antibodies or different oral biologic targeted agents like tyrosine kinase inhibitors, that you could extrapolate from your approval to all these different combinations. And if it worked just as well in the metastatic and early stage setting, you could also extrapolate freely between the two settings. And then lastly, if it works for more than one disease, like trastuzumab for breast cancer or gastric cancer, you could extrapolate between those indications as well. So that is the entire approval process from beginning to end. And right now, in terms of what's been approved so far, the US FDA and EMA have approved these agents with the idea that extrapolation will be used.